Hello students, today we will discuss about osmosis. In the previous lectures of this chapter we have discussed about the diffusion and the rate of diffusion and the factors affecting the rate of diffusion. Today's lecture we will discuss about osmosis and first of all we will discuss about general osmosis, and after that we will discuss about the osmosis in animal cell, and after that in plant cells, and we will discuss how osmosis works and the different terms regarding the water potential. And so let's start. If uh, if we study the osmosis, actually it is just like the diffusion, but the difference is diffusion is a general term that is used for any gas or the liquid molecules can move from the high potential to the low potential area. So in the osmosis, we are not going with the general topic, we are discussing about the specialized material that is only and only water. So let's start. What is osmosis? Actually it is the movement of water molecules from the area of high concentration of water molecules to the area of low concentration of water molecules. So this is the actual difference between osmosis and the diffusion, so don't confuse it. So if a dilute solution is separated from the concentrated solution, actually the dilute solution is that solution in which solute is less and solvent is too much greater amount. And if we compare the dilute solution with the concentrated solution, in the concentrated solution there is always a large quantity as compared to the dilute. Large quantity of solute is present, and solvent of course in the large quantity or the specific quantity is also present. So the concentrated solution here in the dark blue, or we can see the dilute solution is the light blue, and it is separated by the this membrane, and this is membrane is actually the semi permeable membrane, partially permeable membrane, or the selectively permeable membrane. So we can say from the dilute solution into the concentrated solution, uh, the concentration solution is actually the solution in which solute is relative to the dilute is present in the greater quantity, while the water is in the lesser quantity. So we can say by two ways. We can say from the high water concentrated area or high water containing area to the low or less water containing area. This movement is called actually the through the semi permeable membrane. This is called osmosis. Water diffuses across a membrane from the dilute to the concentrated solution, because water is present in this area, in the light blue area high quantity, and in this present water is in lesser quantity. So it is very normal, high from high to the low area through the semi permeable or the partially permeable membrane. So this is the osmosis. Partially permeable membrane is the porous. It means only the selective substances can pass through this membrane, but allows water to pass through more rapidly than the dissolved substances. Or we can say the solute will move in opposite to the osmosis, or we can say the diffusion of the solute molecules is happening at the same time as the osmosis is happening, but in the opposite direction. Since a dilute solution contains in effect more water molecules than the concentrated solution, there is a diffusion gradient which favors the passage of water. It is very easy concept from the dilute solution to be concentrated solution. In the living cells, the cell membrane is a partially permeable membrane, and the cytoplasm and the vacuole in the plant cells contain dissolved substances. As a consequence, water tend to diffuse into the cell by osmosis if they are surrounded by the weak solution. Actually here the weak solution is the solution that contains a very large quantity of water and very less quantity of solute. If they are surrounded by the weak solution, fresh water, if the cells are surrounded by the stronger solution, here the stronger solution actually means the concentrated solution or the solution that contains more solute as compared to the over our cell. For example, the seawater, seawater is actually, we know, concentrated water or the high containing high amount of salts, or we can say the saltish water. The cells may lose water by osmosis. Actually, when we place the cell or the cell containing actually the normal concentration of the salts in our, just like for example, the RBC is only 0.5, 0.85%, 0.85%. So we can say 0.85% if salts amount present inside the cell, and in the environment or the outside the cell, the system will be called as the isotonic system. Actually, the word iso means same and tonic means tonicity or the salinity. So isotonic means the solution containing same amount of the exactly same amount of salts inside or outside the cell. So this is the isotonic system. And when the environment containing salts more than 0.85%, this environment is called as the actually hypertonic environment. So the hypertonic environment is known as the stronger solution containing high amount of salts, for example the seawater. And the hypotonic, actually the environment that contains less salts than 0.85 as compared to the RBCs. So let's see in the detail the animal cell, or we can also say in figure 3.9 that is showing below here, there is high concentration of free water molecule outside the cell than inside, so water diffuses into the cell. So as a result, the cell will be swollen up, or we can see the cell will be become larger because of the entry of water. So this environment is known as the hypotonic environment. So this environment is the hypotonic environment. So this hypotonic environments actually causes the cell volumes increment. So we can say in animal cell, shown very simply, the colored circles represent molecules in the cytoplasm. We can see the colored circles, cytoplasm, and they may be sugar, salt or protein molecules. The blue circle represents the water, and we can see here a very large quantity of blue circle is present. So we can say the cell is surrounded by pure water. Nothing is dissolved in water. It is 100% concentration of water molecule. So concentration of free water molecule outside the cell is greater than the inside cell, and therefore water will diffuse into the cell by osmosis. The membrane allows water to go through the either way. So in our example, water can move into or outside the cell. The cell membrane is partially permeable membrane to most of substances dissolved in the cytoplasm. So although the concentration of these substances inside may be high, they cannot diffuse freely out of the cell because the solute particles are larger than the holes present in the cell membrane. Because it is semi-permeable membrane, the only the solute molecule which are of the pore sizes can pass through and become out of the cell. So there are very less or negligible amount of the solute present that is larger than the hole present in the cell membrane. So water molecule move into and out of the cell, but because there are more of them on the outside, they will move in faster than they move out. So the liquid outside the cell does not have to be 100% pure. As a long the concentration of water outside is higher, so we can say this water become or we can say it become the swollen. And finally, if it is an animal cell, it may be burst because it is not surrounded by the very strong cell wall which is present in the plant cell, not in the animal cell. So as a result of the if we place a animal cell into the hypotonic environment, so as a result the cell will swollen up and finally burst. Water entering the cell will make it swell up, and unless the extra water is expelled in the same way, the cell will burst. Actually, in the human body or in the animal body, there is homeostasis mechanism which controls the extra or the large quantity of water to the entering. So we can say the animal cell cannot burst because of the not too much water level become rise up inside the cell due to the homeostasis. So we can say if the cells are surrounded by a solution which is more concentrated than the cytoplasm, water will pass out of the cell by osmosis and cell will finally shrink. Actually, in the hypertonic environment, the cell will shrink, of course, and excessive uptake or loss of water by osmosis may damage the cell. So, for this reason, it's very important that cell in animal body are surrounded by a liquid which has the same concentration as the liquid inside the cell. The liquid outside the cell is called the tissue fluid, or we can say it is the interstitial tissue fluid, or simply we can say the environment inside the human body, the environment outside the cell, and the cytoplasm concentration of water inside the cell the same. So we can say this condition is called as the isotonic, and its concentration depends on the concentration of the blood. In vertebrates, uh, like the animals or the organisms which contain the vertebrae, the concentration of blood is monitored by the brain and adjusted by the kidneys. So by keeping the blood concentration within the narrow limits, the concentration of tissue fluids remains more or less constant, and the cells are not bloated by taking in too much water or dehydrated by losing too much water. So there was the specifications or the homeostasis mechanism that controls the entry of water inside or outside the cell. But here in the plant cell, there are no too much homeostatic actions, but in plants there is an extra organelle that is called as the sap vacuole, or the water vacuole, or the contractile vacuole, which controls the actually the higher quantities of water if entered into the cell, it may be adjusted accordingly. Plant cell, the cytoplasm of the plant cell and the cell sap in its vacuole contains salts, sugars and proteins which effectively reduce the concentration of free water molecule inside the cell. The cell wall is freely permeable to water and dissolved substances. But the cell membrane of the cytoplasm is partially permeable membrane. If the plant cell is surrounded by water or the solution more diluted than its contents, or we can say if we provide the hypotonic environment, water will pass into the vacuole by osmosis, of course. And the do you know what is the name of the cell membrane of the vacuole? It is called as a tonoplast. The vacuole will expand and press outwards on the cytoplasm and cell wall. The cell wall of the mature plant cell cannot be stretched, so there comes a time when the inflow of water is resisted by the inelastic cell wall. So finally, up to a certain limit, the cell wall can adjust the extra entry of water by the contractile or the sap vacuole, but to a certain extent. After some time, the extra water, even though there is larger quantities of water, but after a specific point there will be no entry of water inside the cell. Since there is effectively a lower concentration of water in the cell sap, water diffuses into the vacuole and make its push out against the cell wall. We can say there is increased contractile pressure inside the cell. This has similar effect to inflating of a soft bicycle tire. The tire represents the firm cell wall. The floppy inner tube is like the cytoplasm, and the air inside corresponding to the vacuole. If enough air is pumped in, it pushes the inner tube against the tire and makes the tire hard. When the plant cells have absorbed a maximum amount of water, they become very rigid due to pressure of the water pressing the outwards on the cell wall. The end result is that the stems and the leaves are supported. If the cell loses water, there is no longer any water pressure pressing outside or outwards against the cell wall, and the stems and leaves are no longer supported. At this point, the plant becomes limp and the wilts. When there is too much extra exit of water outside the cell, or if we place a plant into the salty environment, so this will happen and the plant wilting will ultimately results. So this was all about today's lecture. In the next lecture we will discuss about the uh, how osmosis work in extra detail, and the term water potential we will discuss, and the importance of water potential uh, and osmosis in uptake of water by plants. So this will be our next topic, and what is the importance actually. So today's lecture this was all about, and thank you very much for listening me for almost 15 minutes, and try to grasp all the concepts that I am delivering to you very interestingly. And please do subscribe the channel and press the bell icon for the further important lectures. Thank you.